Hello, please. We plead for everybody. Please try to mute yourself because we're trying to mute everybody here. We are experiencing some technical issues. So please, the moment you are in, try to mute yourself. So try to mute yourself the moment you are in, okay? Thank you. Please kindly mute yourself. Please. Okay, so we're starting very soon. I welcome everybody to Genomac Up. Please, can you mute yourself? It's very, very necessary you mute yourself to avoid distraction. We're trying to do the setting here so that we can mute everybody and give you permission to unmute yourself when the time is right. Please, who is this? Can you mute yourself? So we're going to start very shortly. Please, can you hear me? Do I have any background noise? Hello? Do I have any background noise?
No.
Okay. So I welcome everybody to today's webinar session where we're going to be doing quite a lot. And then we're going to be showing you some practical illustrations on NGS data metagenomics techniques. Okay. And then we're going to open a door for you to gain access to numerous numbers of priceless bonuses, right? Priceless opportunities. So we really need to mute everybody here. Please give me a minute. Can you see here? From here, everyone. Can you see? Hello? Please, we're trying to check if you can mute yourself or not. Please. Hello, can you hear me?
We can hear you. We can hear you.
Hear you clearly.
All right, let's just go this way. We try to work on this later because we just want to try so much to avoid distraction. But in any case, let's proceed. So once again, I welcome everybody to Genomark Up, where we'll be taking you through the entire process and insights, information concerning NGS data metagenomics techniques. And then I hope by this session, and I'm sure by this session, you definitely have quite an information, right? Detailed information on what NGS metagenomics is, the practical applications, the research areas. And if you are a PhD student, definitely we're going to take you through some step-by-step process on how you can actually explore this research in case you want to do that as a research project in your PhD, right, in your program. So we are going to take you through that process. Myself, I'm going to take you through that process. Let me say that. So I'm going to share my screen now. I'm going to share my slide. Can you see my screen?
We can't see your screen. We can only see your face.
What about now?
Not yet.
Yeah, we can see your screen now.
Okay, awesome. Thank you so much. So, once again, I welcome you to this webinar. I'm Oluwaseyi Olawale, the founder and CEO of Genomark Up. So, I'm going to introduce to you what we are doing at Genomark Up. So, we actually integrate creative thinking, innovation, and biotechnology to provide sustainable solutions to contemporary health challenges using bioinformatics, artificial intelligence, and medicinal chemistry. All right, so this is some of the major things we do here at Genomark Up. So particularly, one of the important projects we have currently is that we're exploring African medicinal plants to provide sustainable solutions to health problem. And then we have three aspects of medicinal products we are currently developing. And the first one is diabetics medicine. The second one is diarrhea, and then PI, that is hemorrhoids. So we actually discovered two plant combinations that we tested on patients traditionally, and it was proven to work, right? Because this time around we're talking about traditional testing of the plant, right? So we tested it on about more than 200 patients, and some of them, or quite a number of them, who are diabetics patients, right, testify. We actually saw the medical records shows that those medicine works. So currently we are using bioinformatics, artificial intelligence, and medicinal chemistry to explore the plant, and then we are developing it right now at our lab. So I'm just trying to make you understand some of the things we are doing. So just to put your mind at rest that we are in good hands. So, and we are not only exploring African medicinal plants for drug discovery process, we are also exploring microbes. So that means whatever we're going to do today is actually what we are doing. Okay, just a few months ago, we were doing a particular research project, and we get to discover, I mean, we actually discovered a medicinal, we actually discovered sorry, I'm just trying to admit somebody here. Okay, it's weekend, so that is why. In fact, it was yesterday I discovered that the webinar is today, so I thought it was Monday. So quite a number of different things. Okay, so as I was saying, a few months ago, we were exploring metagenomics techniques or metagenomics samples. We're exploring clinical samples, human gut microbiome samples. We're exploring soil, water, and ocean water metagenomic samples. And the aim of we actually doing that was that we are actually looking for novel peptides that we can use as antibiotics, agents, right? Antimicrobial agents that can be used as antibiotics, to treat staphylococcus aureus. Okay, and also we were also exploring the metagenomic samples in order to discover novel peptide that can serve as anti-cancer agents. Now, during the course of our research, we get to discover some novel peptide, okay? Because we're actually using artificial intelligence. We're using machine learning models, okay, to do some of these practical applications. So we discovered this novel peptide, and currently those peptides are under development. So we actually transiting from the in silico, in silico. Please, can you mute? We really want you guys to mute in order to avoid distraction. So we're actually currently transitioning from the in silico research down to the wet lab research, where we do some experimental verification and then we go to animal trial. Okay, so this is some of the things we're actually doing when it comes to microbes. And like I said, while I was speaking, I said we were actually exploring metagenomics, right? And we will be talking about metagenomics. We'll be talking about what is metagenomics, right? We'll be talking about the research and applications of metagenomics, and then we will show you a practical workflow, right? Bioinformatics workflow for big data analysis when it comes to metagenomic sample, different kinds of metagenomic sample. So I'm gonna take you through that right now. So I hope you can see my screen once again.
Yeah, we can see your screen.
All right, thank you so much. So now, once, like I said before, I'll be talking about the research and applications of NGS metagenomics in bioscience. Okay, the research and applications of NGS metagenomics in biosciences. So what do you imagine as a life scientist? So because you've been here, right, on this webinar shows that you are interested in microbial research, right? You're interested in microbial genomics, you're interested in metagenomics, and that shows that probably you are a life scientist, or probably you're a computer scientist, but you want to base maybe probably your career, master's or PhD, you want to actually streamline your niche to explore genomics. Probably you want to be developing algorithms, you want to be developing tools, okay, for bioinformatics analysis. So, that means in terms of computational biologists, right? So this particular session, this webinar is also for you, okay? So what do you imagine as a life scientist? What do you imagine as a bioscientist? So, okay, let me just try to break this down. What is your imagination? What is your vision? What is your intuition? What is your aspiration as a life scientist? Okay, why are you a life scientist? Now, permit me before I continue, please, I would like one or two persons to give me an answer, okay? Because I always love my session to be very, very interactive. Okay, amazing. We have quite a number of participants from all over the globe. From all over the globe, we have India. We have people from Europe. Let me just say continentally, right? Africa, Europe, America, Asia. Amazing. So please, my question is, okay, my question is, why do you become okay, somebody say very slow voice, please. Can you hear me audibly? Am I audible? Am I audible enough? Yes, thank you, thank you.
Yes, sir,
Now,
you are able.
yeah, good. I need somebody to tell me that the chat room or you mute yourself. Why are you a life scientist? What is your aspiration, right? What is your aspiration as a life scientist? We would love to know. Can you just share with us, one or two people, very quickly? I want this session to be interactive. Why are you a life scientist? Okay. Yes, yes, yes. Very quickly. What is your aspiration as a life scientist? Yes, yes. Go on.
Yeah, as a life sciences, it's all about the creative of knowledge and we look in our environment, there are a lot of issues that we contend with. So as we become aware of a way of solving those problems, so we try to, as life scientists, I imagine a situation whereby I will use the application of the knowledge I've acquired through the scientific method by asking questions, by probing into the problem that is around me, and using my impact of knowledge to solving those problems in order to give solutions to the society and also as well to communicate and have a kind of a global impartation to knowledge.
Amazing, amazing, amazing. That's a very, very wonderful answer. Thank you so much for that lovely answer. So, as you know, if I should pinpoint some, uh, some of the things he said, he talked about problems, right? Because I can see from the chat room as well. I can see problems contribute to the field of science to advance, science to bring possible solutions to health problems, to solve, food contamination and waste problem posed globally by microbes. So thank you so much, guys. Thank you so much. That's a very, very wonderful answer. Thank you also, the person that just answered. Thank you so much. So now, as you have said, okay, the basis of you being a life scientist is actually to solve problems, especially biological problems. And we've seen some of those examples there as well. Okay, biological problems. Now, think about a biological problem in your community as a life scientist. Now, what do you want to solve concerning this problem, and how do you need? What do you need to create the solution in solving, in tackling the problem? Okay, for instance, let's talk about the pandemic, the COVID-19 pandemic. Now, as a life scientist, the problem is pandemic, is COVID-19, right? Pandemic. Now, the second thing is, how do you want to solve this pandemic, right? Now, to solve this pandemic, the basic things about of the solution is that you want to stop it, right? You want to stop the pandemic. You want to stop the spread of the infection. You just want to find one way or the other to tackle the problem, to stop spreading, all right, or to cure those who are already been affected by it. Now, you will need some specific tools, right? You will need resources, you will need skills, you will need research expertise, you will need experience to be able to create a solution for this. Now, let me tell you something. There is a gap between research and the people and the community. And the gap is the entrepreneurial aspect, the commercial aspect, the industrialization aspect, right? That is the gap between it. So it means you as a life scientist, you must be able to have an innovative capacity to be able to identify problem, solve that particular problem with the available resources around you. So it means you must be aware of your niche. You must be able to acquire as many skills as possible that you need in order to make that vision a reality. And this is what we are about to do. Okay, so because we are actually going to be talking about NGS metagenomics, but how would this NGS metagenomics be of value, be of use? How can it become a valuable resource, a powerful tool in your hand as a life scientist to provide solution, not just to provide solution.
Sorry, the voice is lost.
Hello, please, can you hear me? I just lost connection. Hello, can you hear me?
Yeah, we can hear
Yes.
you now.
Yes,
You
yes, sir.
can continue.
I can hear you.
Hello, please, can you hear me? Okay,
Yeah,
because I
we
just
can
lost
hear
my
you clearly
connection.
now.
Amazing. I'm back now. I'm back. Okay, let's proceed.
Welcome.
So, thank you. So, why is it not connecting? Hello? Hello?
Hello, we can hear you.
Yes, yes, we can
Okay,
hear.
okay. I think I really have a serious network issue. Okay, can you see my screen now?
Not yet. We can
What
see
about
it
now?
now. Yeah, we can see it.
Okay, amazing, amazing. Thank you. So, so I asked a question. I asked a question previously, and my question was, can you quickly tell me on the chat room what pathogenic bacteria are and non-pathogenic bacteria? Very quickly, just a point. Just a point. Pathogenic bacteria and non-pathogenic bacteria. What are they? Please, can that person mute?
Pathogenic,
Hello?
pathogenic bacteria are those bacteria that can cause infection, while non-pathogenic bacteria cannot cause infection.
Amazing, amazing, amazing. Thank you so much. Thank you so much for that answer. So, like I said, all right, pathogenic bacteria are those that cause infections, and those and non-pathogenic are those that does not cause infection. So it means pathogenic bacteria are actually virulent or pathogenic, right? So now, this is microbial genomics for you. Which enable microorganisms to infect a host and cause what? Infections. Okay? And cause infections. That is pathogenomics. Now, pathogenomics deals with pathogenic bacteria. And the next thing we have is probiotic genomics, which is the genomics of probiotic bacteria. Now, this particular aspect of microbial research, right, deals with the non-pathogenic bacteria. Now, probiotics are bacteria that are not pathogenic, right? Now, can somebody tell me an example of probiotics? A probiotic bacteria, very quickly, at the chat room. Very quickly, an example of probiotic bacteria, or any food.
Lactobacillus acidophilus.
Amazing, amazing. But when I say probiotics, what's the first thing that comes to your mind?
Like Neptune, that is
Edible microorganisms.
Yeah, edible microorganisms. So I was expecting somebody would say yogurt.
Yes,
right?
yogurt.
Okay, I was expecting somebody would say yogurt, because I said what is protein. So we have metagenomics. Now, metagenomics is the combination of both pathogenic bacteria and non-pathogenic bacteria, because metagenomics deals with an entire community of microorganisms. Let's proceed. So, NGS data metagenomics study. So what is metagenomics? Now, metagenomics can also be called environmental genomics. Now, we are diving deeply into what metagenomics is, okay? Metagenomics can also be called what? Environmental genomics, ecogenomics, or community genomics. It is the study of genetic material recovered directly from the environmental samples. Please, am I audible?
Yes.
Yes, we can hear you.
So, metagenomics is the study of genetic material recovered directly from the environmental samples. So it means you can get metagenomic samples, right? Or you can metagenomic material, right? You can get metagenomic materials from the soil, from the water, from the feces, from clinical samples, from the gut of human, okay, from the feces of human, okay. You can get it from the ocean. You can get it from freshwater metagenomics. Freshwater body. You can get it from the stream. Okay. You can get the samples from anywhere, right? As far as you can actually recover a genetic material from it. That is metagenomics for you. Okay. Is the study of genetic material recovered directly from the environmental sample. Now, this is the definition. This is what metagenomics is, the study of genetic material. Now, there is a reason why you should study genetic material from the environment. There are applications of studying genetic material from the environment. There are applications. There are reasons. And you know, there are industrial applications while you study metagenomics. There are medicinal applications. There are, you know, when it comes to health, there are agricultural applications as well. So the practical applications of metagenomics to study a specific community of microorganisms, that is metagenomics for you. Now, can somebody tell me what? I already said it. Let me not ask because of our time. I said the other time that genetic component are DNA, RNA, and protein, right? Am I? Hello?
We can hear you.
Okay, good. So the other time I was talking about genetic material, and I said they are DNA, RNA, and protein, right? So if you are getting genetic materials from the environment, like I said, there's a reason. Now this is it. When you do, because this is how it starts. Like if you are to embark on metagenomic research, first you get the samples from whatever site or whichever environment you want to get them from. Okay. Remember the other time I told you they can either be soil, water. Okay. They can either be feces, clinical samples. Okay. They can be pond water, ocean water. Now, by the time you get this sample from the environment, you take it to the lab for culturing. Okay, and then you get the samples down to the lab for sequencing. You the specific area of sequencing after this junction. Okay, by the time this sequencing is being done, okay, you are going to get a whole lot of big data. Okay, because metagenomics is diversified. I'm also going to tell you, but there's an aspect of metagenomics that deals with the entire genome of microorganisms that are present in the environment. So it means the first practical applications of metagenomics is that you want to study the specific community of microorganisms in the environment. So it means that when you get this
Your voice is not clear. Sorry.
There is a reason why you should do this, but this is how the research begins. Okay, this is the specific pathway in this metagenomics research. Then the second thing is known for its
I think the host is having network issue.
Exactly. We need to tell him, and he needs to slow down.
Yeah.
It's known for and agriculture. It shed light into the development of probiotics and so much more. Pardon?
Please, can you hear me?
Hello? Yeah, go on.
Hello, can you hear me, please?
Hello, yes, go on.
Yes,
Yeah, please.
yes, we can hear you.
I think you are having a network issue there. The thing is cutting.
Oh, really?
And also you are too fast. Please, can you calm down a bit, slow down a bit?
All right, no problem. Sorry, I'm trying to see the problem. Please, am I audible enough now? Am I clear?
For now, you are okay.
Hello?
Yes, you are audible.
Yeah, we can hear you now. For now, you're okay.
Okay, please, need a minute. Okay, let me go ahead. Sorry. Can you hear me now? Am I audible?
Yeah, we can hear you.
Okay. Hello? Hello?
Yes,
Hello.
yes, I can hear you.
Hello.
You can hear
Okay,
me.
you can hear me now. Amazing. Hello, I think I'm back now. We apologize for the network problem, okay? It's not our fault. It's the network providers. Hello, are you with me?
Yes, we are with you.
Yes, yes,
Thank you so much.
yes.
Thank
You're
you
audible.
guys for your patience. Okay, so let's proceed.
Yes, you can go ahead.
Yeah, thank you, thank you. So I was
Anyone
saying
else?
something. Yeah. So I was talking about the applications of metagenomics, right? So first of all, I talked about how you can start with metagenomics. So the first thing is you get a sample from the environment. Now I said the other time that the metagenomic environmental sample can either be the soil, the water, the feces, or the clinical samples. All right. So as far as you can
Yeah,
actually
we
get
got it.
genetic materials from any environment, you can actually embark on metagenomics research. So the second thing is the second idea about metagenomics when it comes to the application is that you want to explore the entire community of microorganisms that is present in the environment. All right, because you see, in the environment, especially in the soil, there are lots of microorganisms there. You could find virus, you could find fungi, you could find archaea, you could find bacteria in the soil. So, but when you are doing metagenomics, you are widening your scope of research. Do you understand? Metagenomics is about widening your scope of research such that you want to discover so many things at the same time. You want to explore so many things at the same time. That is the idea about metagenomics. Now, metagenomics has an advantage than genomics because we have individual genomics. That's whole genome sequence. Okay, genomics. So because whole genome sequence is targeted towards a specific organisms of interest. So it could be a specific bacteria, it could be Lactobacillus fermentum, it could be Staphylococcus aureus, right? So you could actually sequence a genetic material, that's nucleotide sequence, DNA, right? The ACGTs, right? Of the entire whole genome of an organism. You can actually do that for one organism. But you see, metagenomics can do that for the entire organism that is present in that specific environment. That is where metagenomics is so wide. It is like you trying to study a community than studying just one person. Okay, that is the idea about metagenomics. Please, are you following me? Hello?
Yes, yes, go ahead.
Yes, yes,
Yes,
sir.
go
Yes,
ahead.
sir.
Yeah.
We can
I'm
hear you.
actually the PhD student. Sir, I am actually the PhD student. I sent my sample for whole genome. I want to know the difference between whole genome and metagenome. Kindly explore this.
Oh, definitely. We're going to take questions as we proceed, okay? We're going to take questions. So, as I was
Okay.
saying, okay, whole genome sequence is targeted towards a specific, the entire whole genome of just an organism. But metagenomics is targeted also, there's an aspect of metagenomics that is targeted towards the entire, genetic makeup, right, of the communities of microorganisms in a particular environment. So if your research is based on bacteria, you can actually do metagenomics to focus on the entire community of bacteria that is present in that particular environment. Okay, so the bacteria now could ranges, because according to taxonomic classification, right, we have from the kingdom, phylum down to the species level. But you see, metagenomics will actually give you a full detailed information about the taxonomic classifications, the samples that is present in that environment, and it's just like community based. Now the second thing is that it's known for its application in health and agriculture. Now in health, here we have pathogen and plant disease identification. So in health we have pathogenic research. Okay, so imagine, actually, I'm still going to talk more about this, but I just want you to know this point. Disease resistance breeding, this is plants. Disease identification, still in plant. Plant pest control, okay, still in agriculture, right? Weed management, agriculture. Abiotic stress management, agriculture. Post-harvest management, agriculture. Discoveries, so much in agriculture. So the second thing is that it shed light, metagenomics shed light into the development of probiotics and so much more. More so, metagenomics can also be used to do discovery, drug discovery, just like what we are doing at Genomark Hub. We are exploring the metagenomic samples to discover specific anti-cancer agent or antibiotic agent, okay, to deal with some specific pathogenic infections. Okay, now we are going to talk about the applications of metagenomics now, okay, in health. Now listen very attentively, because as we go on, I'm going to give you the practical workflow. Okay, I have the slide that how you can actually proceed from the research level down to you having a research paper, down to you having a result of a specific question you have in mind with metagenomics. But follow me steadily. Now in the applications to health, we have outbreak detection and surveillance. Okay, outbreak detection and surveillance. So it means you can use metagenomics to what? To explore outbreak detection and surveillance in a specific community. Now, let me give an instance. For instance, let's just say a particular pandemic, okay, or a plague or an infection broke out in a specific community, or maybe a community of 10,000 people, okay? A community of 10,000 people, a particular infection break out, okay? And you want to trace the infection down to the source. Okay, for instance, let's just say the infection is actually caused by a specific, a particular bacteria, let's say Staphylococcus, okay? But you want to trace down that specific bacteria you discovered down to the source. Probably you want to know if the source is from the water environment or the soil environment, or if the source is from the clinical environment, from the pond, from the gutter, okay? Probably it's from the food, okay? Maybe it's from the poultry when it comes to agriculture, like animal nutritional or animal agricultural base, right? So this is actually an awesome and interesting area, which is metagenomics, to explore in order to trace that particular infections down to the source. Because why am I saying this? By the time you do metagenomics, you start the metagenomic research, you getting the samples from the environment of that specific environment, you must have discovered a substrate, all right, or a strain. You must have a sequence, or you must have taken a sequence of where the source of that outbreak came from. So it means if it actually came from water and you get the water samples, it means that in that water sample, that particular source of infection or outbreak will be in the water sample. Now, by the time you do sequencing, you get the genetic material, which is DNA, of the entire set of, the genomic makeup of the community of microorganisms in that environment. Now, by the time you start doing analysis, you will discover, or you will know, the specific substrate of that particular organisms in, based on this specific community of microorganisms, because you are going to be given the entire community of it. You're going to be given the name, all right, the strain, the isolates is going to be there. Okay, now by the time you now begin to evaluate your analysis, you would discover that particular strain in the metagenomic sample. I hope is that clear? And as such, you can use this for outbreak detection and surveillance. And you can do this in order to track the spread of infection, or maybe, for instance, antimicrobial resistance. You can use this to track the infection, to track the spread of antimicrobial, antimicrobial resistant genes. Okay, you can use it to track mobile gene elements that are causing that particular outbreak, making it to jump from one individual to another. Okay, making it, to spread from one individual to another. And, by the time it's spreading, the infection could be worsened, could be more virulent. I hope this is clear. Hello? Are you following?
Yes, sir.
Yes, clear.
Amazing, amazing. Now, the second thing is diagnosis. Okay, you can actually use metagenomics for diagnosis, diagnostic techniques. Okay, because in metagenomics, we'll be talking. There's an aspect that deals with the phenotypic and the genotypic aspect of pathogenic organisms. Okay, so it means by the time you do metagenomics, you'll be doing some in silico research analysis, where you'll be discovering using machine learning, where you could discover some specific phenotypic features that is probably contributing to the resistance, the intermediate, or the susceptibility of the specific microorganism that is present in the metagenomic sample. So it means with metagenomic sample, you can actually trace down your source to a specific issue that is actually associated to the problem you're trying to solve. Okay, then we have precision medicine. Metagenomics also can be used to explore precision medicine. Why? Because by the time you begin to explore the metagenomic sample of a particular human, maybe gut microbiome, you could discover the interaction of microorganisms at the gut microbiome of the human. Because you see, human being also has good bacteria in them. Now, the imbalance of this bacteria in human could actually lead to a specific problem, could actually lead to a serious issue, right? In the body of the human, right? It could lead to a particular issue. But studying the entire genetic community, right? Makeup of the genetic makeup of the entire microbial community in human, you could actually discover ways, patterns at which you could narrow down, or at which you could explore in order to provide a specific precisional approach in order to deal with the infection that is affecting or dealing, battling with a specific individual. Okay. The next is drug discovery. I've already talked about drug discovery. Then the next one is nutrition and diet. You can use metagenomics to explore probiotic communities. Okay, you can use it to explore microorganisms, community, in order to narrow down your research to a specific nutritional benefit. Now, let me talk about this nutrition in agriculture. Okay, this nutrition and diet in agriculture. So you can actually use metagenomics to explore the nutritional aspect of agriculture. How? So now, the communities of microorganisms in the soil, like in the farm, right? In the farm, where they cultivate plants and all of this. Now, there are microorganisms that affect the growth of the plant. You know that, right? Am I right? I need an answer.
Yes,
you reply right. The chat. Yeah, there are organisms that could affect, that can even affect the growth, that could even affect the growth of, of the plant itself, right? So, and that is why you should do metagenomics, because you doing metagenomics, you'd be able to actually streamline your research to a specific virus or bacteria that is causing infection on those plants. And as such, you'll be dealing with a very huge problem. All right, you'll be dealing with a very huge problem, and then it will increase productivity and yield. Now, there are specific bacteria that are actually associated, with the growth of plants. All right, maybe the yieldedness of a particular crops or seed in the farm. So you doing metagenomics, you will actually be able to know, oh, it is this particular virus that is causing this problem, and this is the genetic makeup, this is the resistant genes in it, this is the virulence genes in it. So that, okay, and these are the drug target. Okay, can we develop a solution on how to target this, to deal with this drug target so that we can stop this infection? And as such, by the time you have a solution, you are already becoming, an innovative researcher that's creating or providing solutions to the problem that is facing the community. Okay, so you can investigate plant infection, like I said, you can investigate soil and plant nutrition. In fact, you can use metagenomics to understand what categories of nutrition is found in the soil. Processing of agricultural products, because food, based on food spoilage and quite a number of things, food infections, that's poison, I know you understand what I'm trying to say. Okay, now the industrialization and commercialization of metagenomic research. One, we have enzyme found by researchers from metagenomic source can potentially be commercialized. Enzymes. We have drug discovery. We have probiotic engineering, etc. All right, using metagenomics. Okay, using metagenomics. So now, what is the approach of metagenomics? The approach. Now, one, we have targeted sequencing. Number two, we have shotgun sequencing. Okay, now this is just talking about the sequencing approach. If you have your sample, you take it to the sequencing lab. Is either they do targeted sequencing or shotgun sequencing. Now, targeted sequencing is focused on specific genes. Okay, maybe 16S RNA. So that means for metagenomics samples actually use targeted sequencing approach. Okay, the second one is shotgun sequencing. Now, as you can see, shotgun sequencing allow a comprehensive sampling of all genes in all organisms present in a given complex sample. Can you imagine that? So it means when you do shotgun sequencing metagenomics, by the time you start doing analysis, you'll be able to direct your research, right, to, in fact, to explore all genes that is associated with the community of microorganisms that are present in that specific metagenomic sample. All right, so now we have the method enables microbiologists to evaluate bacterial diversity and detect the abundance of microbials in various environments. So the principle here is that it relies on genomics analysis of a sample from a complex environment containing more than one microorganisms, hence providing a virtual composition for this sample. So this is the process. I told you that I'm going to show you the process. So if you are a PhD student, you're a researcher, you are a life scientist interested in this, now this is some of the things you can do. Now listen, this is sample collection, DNA extraction, sample preparation, and sample analysis. See, it is before you can do analysis using metagenomics, you actually necessarily don't need to embark on sample collection, DNA extraction, and DNA extraction, sample preparation. Now listen, this is actually lab work and then sequencing. There are whole numbers of sequences that is found on NCBI database and other databases as well on metagenomic sample that you can get, explore them, and use them for a great benefit. Exactly. So, and we are going to be having a workshop, a practical sessions, okay? A practical sessions where we get real life data from NCBI database, and then take you to the step-by-step approach on how you can, go through the entire research process in order to come up with a valuable result that can be used for publications. But first, the process is actually by collecting the sample from the environmental source, DNA extraction, sample preparation, which is the sequencing aspect, then sample analysis. So the sample analysis is what the workshop is all about, is what the practical applications is all about, okay, which we are going to be introducing you to. Is that clear? Hello?
Yes, sir.
Yeah, we can hear you.
Continue. Great. So the
Yes,
steps.
we can agree.
Pardon? So I'm going to take your questions as we proceed. Now, the steps involved in the analysis of metagenomic WGS sample. Now, this WGS sample is not actually whole genome sequencing. This is all shotgun sequencing. Okay, Whole genome sequencing is different, specific to just a sample. Just like somebody has a question, what's the difference between metagenomic and whole genome sequencing? Whole genome sequencing is specific to just an organism, but metagenomics all organisms. But we have different approaches. Okay, remember I told you the approach is targeted sequencing, and the category of targeted sequencing here is 16S metagenomic sample. You can search that on Google. Now the second one is shotgun metagenomics, which is this WGS I'm showing you here. Okay, the WGS here, which is whole shotgun sequencing. Okay, whole shotgun sequencing metagenomics approach. Okay, so the first step whenever you want to carry out your analysis, okay, the first step is assessing metagenomic samples on NCBI database. Okay, assessing metagenomic sample on what? On where? NCBI database. On NCBI database. Please give me a minute. Hello, please, is there anybody with question before I proceed? Hello? Anybody question?
Yes, we can hear you. Yes,
Yeah,
we can hear
anybody
you.
with question? Before I proceed. Anybody with question before I proceed? I think there's a question. Let's proceed.
I think it's better you finish before we ask questions.
It's all right. It's all right. So let's proceed. It's all right. Now, the steps involved in the analysis of metagenomic sample. The first one I said is assessing metagenomic sample on NCBI database. Okay, this is the methodology. This is the practical workflow. Okay, practical approach. Assessing metagenomic samples on NCBI database. Or you get your sample, you take it to the lab, you do the DNA extraction and prepare the sample. Okay. Now the second step is that you need to work with pipelines for metagenomics analysis. There are different pipelines. There are different softwares. But I'm going to show you one of the software today and show you some of the tools you can use for metagenomics analysis on the software. Okay. So just stay put. Okay. Introduction to pipelines for metagenomics analysis. Then we have raw-rich processing and genome assembly. Raw-rich processing and genome assembly. Okay, raw-rich processing and genome assembly. Then we have taxonomic classification and evaluation of results. Let me start again. Raw-rich processing and genome assembly. Access to pipeline tools for the analysis. Now, after you've gotten tools, you've discovered, okay, this is the tools I want to use. You need to do rich processing and genome assembly. Reads processing, you need to process those reads. Why? Because there could be some errors in the reads. There could be errors. There could be places where there could be some sequential errors. Okay, so you need to correct those errors. That's why you reach processing, and then you assemble them, because what you are getting is raw sample. It's samples that maybe it's up to 3 gig, 4 gig. Okay, 4 gigabytes. But by the time you do genome assembly, you'll be able to rearrange the framework of the genome, the entire genome sequences, and then create a consensus context, okay, or consensus sequences which are context in faster format. Okay, the raw reach will always be in FASTQ format. Then we have the taxonomic classification and evaluation of results. Okay, where you do the taxonomic classification talking about biodiversity. Have summarizing comprehensive result. No, okay, we have the comprehensive metagenomic analysis. At the comprehensive metagenomics analysis, you'll be doing genome annotation, you'll be doing gene prediction, you'll be doing specialty gene prediction, specialty gene category, you'll be doing specialty gene evaluation. Okay, that is the comprehensive genomics analysis, where you'll be talking about virulence genes, mobile gene element, you'll be talking about antimicrobial resistant genes using shotgun metagenomics techniques. Okay, comprehensive metagenomics analysis. That is what it's all about. Virulence genes, mobile gene element, some particular genes that are also associated to metabolic pathways. Okay, metabolic pathways, some that is associated to lipid, carbohydrate pathways, some of this, go annotations and some of these things. So that is comprehensive metagenomics analysis, where you'll be focusing on the genes that are encoded by the entire DNA structure that is found in the metagenomics entire sample. Okay, then we are summarizing the comprehensive result. This is where you'll be having visualizations of result. You'll be arranging your result. You'll be narrowing down, streamlining your research to a specific genes of interest that are interesting to you. Okay, they're interesting to your research. They're interesting to the problem you're trying to solve. Then we have evaluating the antimicrobial resistant genes. Right? I've already talked about this. Evaluating the virulence genes, the mobile gene element, computing and evaluations of results. Okay, so this is the steps when it comes to shotgun metagenomics analysis. We also have other areas like chromosomal plasmid sequences. Okay, though I didn't list it here, but it's also available. Chromosomal plasmid sequences. We also have other areas like network analysis of the metagenomic sample, how virulence factor is connected to antimicrobial resistance, connected to the taxonomy biodiversity, mobile gene element. So metagenomics analysis is so wide, and one of the reasons why you could actually use metagenomics analysis in clinical research or medicine, or biomedical research is the use of the antimicrobial resistant genes and the virulence factors. Okay, because you want to study specific virulence factor contributed to the virulence outbreak of this specific infection or a particular infection in the environment. Okay, so this is the NCB I was talking about. Introduction to pipeline tools and applications, accessing real-life data samples from NCBI sample, archive, and doing some other stuff. Okay, then we have rich processing and genome assembly. We have the FASTQ, which means you have to treat. We have the genome assembly. You have to rearrange the entire genomic structure. Okay, you have to put them together. We have the taxonomy classification and evaluation of results. Now, this is what I'm talking about, the taxonomy classification. Okay, can you see the roots? Right? Are you there, guys?
Yes, sir. Yes, sir.
Yeah,
Amazing.
we can see it.
So this is taxonomy classification, where you do the entire genomic diversity. Now, can you see the root? Here's the bacteria, like the kingdom, phylum, all right? The family, genus, down to the species
Sir,
level.
excuse me. 1 minute, please stop. We can get this from the metagenome or whole genome?
No, metagenome, not whole genome. Whole genome is just one organism. If you do this kind of analysis for whole genome, you only have one name of an organism. But when you do metagenomics analysis, you'll be able to see various numbers of bacteria, like microorganisms that is present in your sample, be it virus, be it Homo sapiens sample, be it fungi. It could be eukaryotic, it could be bacteria, it could be archaea, right? You would definitely get them all, and then you will see the names. You see sometimes you see the substrains, right? You see all of them. So did I answer your question?
If we want to get the only one bacterial genome population, then what we can do, either whole genome or metagenome?
One bacteria?
Only one bacterial population.
Yeah, you have to do whole
know.
No, you have to do whole genome.
And then after a whole genome, we further know about the taxonomic classification.
Yeah, it is. No, you can actually do taxonomic classification in whole genome, but when you do that, you only have one name. You only have the name of the sequence, just one. You will not have it this way because, like I said from the beginning, metagenomics deals with the entire community of microorganisms present in a sample. So you have the names of different microorganisms that is present or organisms that is present in your metagenomic sample. Exactly. So if you want to study a bacteria, go with whole genome. But if you want to study many bacteria or you want to narrow down your work to a specific bacteria, okay, you can do metagenomics. But it will be, if you have a specific bacteria in mind, it's advisable you go with whole genome. But if you want to broaden your research and study maybe 4 different host pathogenic bacteria at a go, you do metagenomics because you'll be discovering genes that is associated to this particular bacteria that are invasive to the human as a host or to animals or to plants. Is that clear?
Okay. We can know the virulence factor and resistant gene in the metagenome, sir.
Yeah, you can also know that.
I am correct, sir.
Yeah, in metagenome, you know that. You can also know that in whole genome. But when you do that for whole genome, it is only for that bacteria. But when you do for metagenomics, sample, it is associated to entire bacteria or specific multiple bacteria that is present in that specific region. So
Okay, now clear. Thank you.
Yeah, let's proceed. So we have the comprehensive metagenomics analysis, which I've already explained the other time, right? Which I've explained the other time. So and we talked about antimicrobial resistance. Okay, we've talked about that. We've talked about so we said is the ability of bacteria to resist the effect of antibiotic drugs that are commonly used to treat them.
Can you pipeline again, please?
Please, when I finish, can you ask your question? Let me finish first, okay?
Okay.
Yeah, thank you. So that is antimicrobial resistance, okay? So the ability of bacteria to resist the effects of antibiotic drugs that are commonly used to treat them. That's antimicrobial resistance. But you see, by the time you start exploring metagenomic samples, you'll be able to discover genes, specific genes of interest that are resistant to the bacteria. You will know the antibiotics that the genes are resistant to. Because here we're talking about genotypic profiling of antibiotic resistance in metagenomic sample. So, and then you get to understand the effects of these things on them. Then we have the resistance arises through one of the following: natural resistance, genetic, and acquired resistance from another. So, but this time around, when you're doing metagenomic analysis, you'll be able to explore the AMR phenotype, AMR genes, the antibiotic, the resistant mechanisms, the gene products, the shared genes as well. Okay. Then we have the virulence or the pathogenicity determinant, which is the virulence genes or virulence factor, which facilitate the successful colonization and survival of bacteria or cause damage to the host. This is virulence factor, and very, very important part of metagenomics analysis. Then we have the mobile gene element, segment of DNA that encodes enzymes and other proteins that enable the movement of genes within a genome and outside genomes. Okay, so this is mobile gene element. We're also going to do that. Now, the practical sessions. There is a software I'm going to show you. This is Genius software. Can you see? You can actually download it. Okay. I could provide a link for you to download the software online, but you cannot activate it. You need to buy the activation key, and the activation key costs $400. But we have planned to give everybody who will be part of what we are about to show you, what we are about to open you to, the opportunity that is behind this webinar. Okay, we're going to show you all of that. But see, we are going to give you the free activation key for life to activate this particular software tool. This software tool can do transcriptomics analysis, genomics analysis, metagenomics analysis, protein, primary structure, protein design, predictions, and variant sequence analysis, where you try to study cancer genomics, understand variant point mutations, where you can do virus analysis. When it comes to virus genomics analysis, you can actually use the software. It is widely used, widely known, because it has in it a whole lot of tools that you can use individually, embedded in it. All right. And we are going to be showing you how to use the software for different kinds of analysis in the next slide I'm going to show you. But meanwhile, hello guys, are you with me?
Yes, we are with you.
Yes, sir.
Yes.
I mean, thank you. So, like I said, now, for instance, if you want to do microbial analysis, okay, if you want to do microbial analysis on this particular metagenomic sample, okay, as you can see here, we have a whole lot of samples. This is like a metagenomic sample. This is like a raw data, okay, that is present here. So you can do taxonomy classification with this software. Taxonomy classification, like taxonomy diversity, understand the community of microorganisms that are present in the metagenomic sample. So all you need to do is click the sample. But okay, let me show you how you can import it. So once you have the software, okay, you go to file. When you go to file, you import from file. So click. So if you have the metagenomic sample, so just pick the sample like this, okay, DNA sample, and then upload. So you can create sequence list. So by the time you do that, you have the whole numbers of samples that is present in it. So, but that is if you have a metagenomic sample, you import it. So this is like a microbial sample here, and then you go to tools, and this is 16S biodiversity or classification of sequences. Please, can I finish before you ask your questions, please? Okay.
From where we got this, Vayan?
Please let me finish before you ask your questions. Okay. So then we have the 16S biodiversity. So by the time you click 16S biodiversity here, you'll be able to submit your sample. So let's say genomarkup.com. Okay. No, sorry, email at gmail.com. Then you submit. So I think there is an issue here. The sequence, one of the sequence has a longer read than 100 than 10,000 nucleotide sequences. So let's use this. I just want to show you as an example. Okay. So now you click 16S metagenomics. You click this and then you submit. Now, the moment you submit, you proceed and it's going to run the analysis. And by the time the analysis is done, so I think there's a problem communicating with 16S web server. So I think the problem is from my network here, connecting to my software. But when you do that from your end, you'll be able to do it. So, but this is, I just want to show you that you can actually use this for 16S biodiversity. But this is just one of the softwares. We have different pipeline tools for such kinds of analysis to do taxonomy classification, like the one I showed you previously here. Okay, like this one that I showed you here. All right, like this specific one. You can use the software, but there are other softwares that we are also going to give you, show you how. There's other online tools we're going to show you for nanopore reads, all right, and different kinds of analysis that you can run for metagenomics and projects. Okay, so now let me take you to the practical session. Okay, so I proceed, is there any question? Any question before I proceed, please?
Yes, please. I have a question.
Thank you.
Can you hear me?
Yes, yes.
Yes, please. Currently now I have some samples in the field. Yes, a field trial, and I noticed that there are some bacteria infection on the field. So there's no information on those bacteria that infect the plant. So if I'm to carry out a metagenomics analysis to identify those bacteria now, will I do metagenomics or what? I just need your advice.
Okay, so hello, can you unmute again? Can you ask your question again? Sorry.
Okay, can you hear me now?
Yeah, ask your question again.
Yes, I have a field trial now. I'm working on a sample, is in the field currently, and I noticed that there are some bacterial infections on the leaves. Then I don't have the information of those bacteria, and currently online there is no any information on them. So to identify them now, will I do metagenomics or what?
Are you now? Let me ask you a question. Are you dealing with a whole community of bacteria?
Yeah, currently I don't know if there are more than one or different bacteria. There is no information currently. So just to identify them and do a taxonomic study, what should I do?
Now, if you are working with a particular sample and you believe that there are a lot of organisms in them, right? There are a lot of bacteria in them. You can do metagenomic sample. The thing is, you can actually you just need to get a sample, do the culturing, and get acquainted or get a partnership with a particular molecular lab. Okay, so and then after the culturing, they do the sequencing. So that is if you have a target, that is if you know very sure, okay, that the sample you are actually getting contain different numbers of bacteria. Now, this is something else, okay? This is another thing. Are you with me?
Yeah, sure.
What microbiologist does, okay, to actually get a specific sequence, okay? To actually get the whole genome sequence is that they try to streamline their culture only on specific bacteria. So, they apply some reagents, some substances in order to kill or to stream out, right, every other kinds of organisms. So it means that what you'll be doing is that you'll be working with everything at a whole, because that is metagenomics for you. Take it to the lab, or you just try to have a partnership with the molecular lab. They will take you through the process. Okay. So now, based on what you're trying to do, if you believe that there are more than one or lots of microorganisms that is associated to what you're doing, then you can go with metagenomics. But if you know that it's just one, I would advise you go with whole genome. Do you understand?
Okay.
Do you understand? I would advise you go whole genome. But if you know that you want to target more than one, you want to target more than two, more than three, that are associated to different infections in the environment, then go with metagenomics. Exactly.
Okay. Okay, thank you.
Yes.
Can I just add something there?
Okay, please go on.
Hello.
Yes.
Yeah. The question I had wanted asking him is, how did he know that is actually bacteria? Because I'm looking at it, it's likely to be fungi or virus.
That's another thing. So you must know what you are actually working on, or what you want to work on. You must be sure of what you are doing.
You say you are working on a leaf, on a plant, isn't it?
Yes.
Please go on, Isaka.
Yes.
Yeah.
Yes, I'm with you.
Hello.
I can hear you. Yes, I can hear you. Please continue.
Yeah. You say you are working on a plant, so it's likely to be fungi or a virus. So you should be sure, because if it's fungi and you are targeting bacteria, you will not get anything.
Yeah, thank you so much for that.
Another thing.
And can I say something?
Wait, before you say something.
Go ahead.
Please let me say something before you speak. Okay.
Okay.
There are bacteria. There are actually bacteria that affects plants. Who's hello? The person that
yes.
Ocon. Yeah. So there
Hello,
are.
can I speak, please? Hello. Hello. Can you hear me?
Yes.
We can hear you. Go on.
Yes.
Go on.
Okay, I'm Dr. Kenneth Okon. Right. Is metagenomics, for those of us from developing countries, are too expensive for you to undertake. So if you are in the lab as a microbiologist and trying, you must be targeted to what you actually want. Because if you don't know, naturally you cannot undertake sort of venture. And again, it's easier for you in virology and environmental to adopt metagenomics. For those of us in medical microbiology, we know what we want. Actually, that is bacteria. And when we go on, maybe where metagenomics fits into us. Look, for instance, One Health concept, where we want to look at relationship of antibiotic resistant gene. Where is it coming from? Where is it we're able to track for public health? But one thing remains, and that is the cost of most of this research. At the end of the day, you find that you won't be able to look at it. So whole genome sequencing still result, but we can still do if we have those facilities. Thank you.
Yeah, so now please let me come in. There are bacteria that actually affects plants. Okay. We're working with one of our clients. She's from Belgium, and there's this particular bacteria that causes a particular disease in plants. And she got, I think, about 10 of the whole genome sequence, from different areas. And that particular bacteria actually caused infection in that particular plant, right? So it's not, I just, from all that we have said, I want to pinpoint something. So there are bacteria that also causes plant infection, okay? So I just want to clarify that. Now,
Yes,
based on
actually, sorry, we are not saying that there's no organism of bacterial origin that can cause it, but I want him to be sure before
exactly.
he go ahead.
Exactly.
Yeah.
Exactly.
Yeah,
Now,
I got that. I got that. Thank
now,
you so much.
now,
Can I
sorry,
say
sorry.
something?
Okay, go on, go on, go on.
Yeah, thank you so much, Doc. This is a project, and some people have already worked on the identification through morphological parameters and all that. So I'm just
Amazing.
coming in to identify using the metagenomics. That's the reason why I asked the question. Is that we
Listen,
have the
if you want
information
to
now as
identify,
bacteria.
okay, Isiaka,
Yeah,
if you want to
yeah.
identify, okay, I would suggest you do ITS sequence. ITS sequence is Sanger sequence that is targeted towards a specific gene, and it's not costly. Okay, it's not costly. If you are from Nigeria, it should cost you around maybe 20,000 or maybe 50,000 naira. So for you to get the ITS sequence, and then you do molecular identification through BLAST on NCBI database, and you will know the name. I'm just giving you the practical workflow now. Do you understand?
Okay.
So
yeah,
it's very cheap. But when you are talking about metagenomics, it is that you want to study a whole entire sample. And if you want to do that, your sample must be coming from the soil, the water, the clinical, the gut, or the gut microbiome, even from food. But the idea is that you are actually doing this research in order to explore a whole lot of samples together at a go. Do you understand it now? So
okay, thank you.
Good. So, and as touching in clinical, like he said, it can also actually be used for outbreak detection. It can be used for surveillance. Like what I said previously, it can be used for surveillance, because by the time you're doing that, you are actually trying to figure out mobile gene elements that is common to different microorganisms that is present in that specific region, right? That is present in that specific region. So even in clinical, right, you can explore metagenomics. And while you are doing that, you are actually having access to a whole lot of microorganisms at the same time. Because by the time you started investigating the genes that is in microorganism in whole genome sequence, you could find it there. So it means genes in the whole genome sequence of a bacteria could be found in metagenomics. Why? Because those components from the individual sample is also found, that particular individual sample, like the genetic component of that particular individual sample, is also found in the metagenomic sample. And do you know something? WHO even gave an insight into a more advantaged level of metagenomics than whole genome sequence. Now, this is it. Whole genome sequence at times is even more costlier than metagenomic sample. In Nigeria presently, you can do shotgun metagenomics sequencing for like 150,000 naira or 200,000 naira at Inkaba. So, and there are some levels of whole genome sequencing that is even more costly. So this is it. So I know you guys still have some questions, right?
Yes, I have a question on my own.
Yes, sir. I have one more question.
It's not actually a question, but clarification.
Okay.
Hello. Yes. I'm just looking at the whole genomic sequencing and the metagenomics in terms of surveillance.
Amazing.
If you look in terms of surveillance outbreak.
Okay.
I'm just looking at how will metagenomics identify an outbreak compared to whole genomic sequencing? Because if you look at the coverage in terms of read length, whole genomic sequencing is better.
Actually, I get you.
Yeah.
Listen. This, what you just said now, is actually a challenge. Do you understand? But
yes,
we are having advanced technologies. Are you getting it? That can actually increase the coverage read of the entire whole genome sequence in a sample. Haven't you seen a metagenomic sample that is about 30-40 gig? Just one.
Okay.
40 gig. Imagine if a metagenomic sample is 40 gig. So it means that the actual read length is so long. Now, this is another thing you should know. This is another thing you should know. Do you know that certain bacteria have similar or close range genetic composition?
Okay, yes.
Do you know that? Yeah, because when you compare Staphylococcus aureus and maybe Lactobacillus fermentum, you could find 60% similarity. You could find 50% similarity, right? In fact, you could even find some genes that is present in Staphylococcus aureus in fermentum. Right? Are you getting me?
Yes, yes.
Yeah, so these are some of the techniques that is behind metagenomics. These are some of the techniques, but there are advances. The reason why it's more advantageous, right, is because of you can actually study a whole different samples that they go. Now, we had a sample, a client from China who was doing his PhD on freshwater metagenomics. While we are doing this analysis, we get to discover that some of the genes we had a specific description of the organism it came from in the metagenomic sample. Do you get that right?
Yes, yes.
Yeah, in the metagenomic sample, it's there. In the practical courses that we're going to take you through or introduce you to, right? It's there. Such that when you do analysis, you will see it. It will be classified there that this particular gene is resistant to this antibiotics class, and this is the organism is associated to in the metagenomic sample. So there are advances, but when it comes to surveillance, because actually in Genomark, we are doing surveillance project for Africa. We are exploring the entire set of public access data samples in Africa presently for surveillance capacity to build a software tool for Africa surveillance project. Right? So I get you very well, but we are not using metagenomics. We are using Ogenome. That's the support. That's to support your claim.
Okay.
Do you get it?
Yes. Yes.
Yeah, that is to support your claim.
I was actually saying it because I know that metagenomics is cheaper. If I can use it for surveillance, why going for whole genomic sequencing? That's what I'm looking at.
Exactly, metagenomics is cheaper. Exactly. So but then it has its advantage, really. It has its advantage.
Yeah.
Some of the advantages, one, the community. You will know the organism. You will know that, oh, this organism is present in the environment. Oh, people are shouting COVID-19 in China, COVID-19 in China, and they say that, oh, there's COVID-19 in Nigeria. In that particular environment where the person is living, go and get the soil sample. You could find it there. I don't know if you understand what I'm saying. That is to show you, because when you look at the taxonomic classification, it will show you as proof that, oh, there's COVID in it. In this particular environment, there's this particular virus in this particular environment. Okay, for instance, if a plant is dying, you want to investigate the soil. What is the virus that is causing it? You do metagenomics analysis. Are you getting it?
Hello.
Yes.
Can you hear me, please? Can you hear me?
Yes, yes.
Yes, sir. Yes, sir.
Yes.
I
know your voice is breaking. Yeah.
You can hear me now?
Sir, one more.
Yes, yes, go on.
Yes, we can hear you.
Okay, me for one word. Okay, is she on? Okay, I can continue.
Continue.
Sir, I am working on the biofilm of the wound infection. So
Please,
I send my.
I will listen to you guys. Hello. I will listen to you guys. Let's take it step by step, okay? I'll definitely listen to you guys. Go on.
Sir, I am working with the biofilm of the wound infection. I send my sample for the whole genome, but by the pooling from the different region, like the foot region, from the leg region, 2-3 samples from the foot region, is it all right to do pooling by the whole genome or metagenome?
Oh, you want to pull the fragment, the DNA fragment from different regions as whole genome? Is that what you're saying?
Yes, sir.
Well, it's possible, but then, oh my God, that is technical. That's technical, right? That's technical. At times, you might not find the full coverage of what you're actually looking for. Do you understand what I'm saying?
Okay, sir.
I don't know if I really get your question, but that's it. Okay, that's it. So,
Hello.
Hello.
Can you
yes,
hear me?
yes, go on.
All right.
My question
What?
is, I'm working on a biocontrol agent against a fungal pathogen. So my question is, can I use metagenomics to check the efficacy?
Please, can you ask your question again? Can you ask again?
I'm working on the efficacy of a biocontrol agent in the management of a fungal pathogen in rice.
Well, let me ask you. Can you send it? Can you send it to me? Because can you? Can you just type it? I can't hear you. Can you just type it? The next person before you finish typing.
Hello. What I'm saying is, if you look at the trend of publication, there was a late publication from Denmark, which was populated this last year, that has to do with sewage and running water from 101 country. Is it metagenomics better in environmental sample? That paper dealt with sewage and running water, and there was another paper from US where they take samples from 23rd places, train stations. So when you look, I'm not saying that it's not applicable, yes, but it is better appreciated when you are dealing with the environmental and the tracking surveillance you practice. Then you will see that which has been a very good thing.
Yeah, that's true. That's true. Okay, please, can you permit me? Let me just try to proceed to the practical introduction, then we take the next questions, okay? So, now the practical applications is this. So, it's interesting that so many people have left. Now, some people have left already, but that means they have left this, they must have missed this opportunity. Okay, so now, for you to be able to do metagenomics analysis or research, okay, especially when exploring the pathogenic aspects, okay, we have three things available that we want to show you. The first is fundamentals of bioinformatics. Okay, with fundamentals of bioinformatics, you have basic idea about bioinformatics. Like the person that was talking about molecular identification, you need these fundamentals of bioinformatics for ITS sequence. Okay, and then we are actually giving this out at a very discount price. Okay, as you can see here, we have it to be 50%, $50 for fundamentals of bioinformatics, but right now we are offering it at $6 with based on coupon code, that is at 88% discount price. And then we'll give you a paid software, bioinformatics software, which is what I just showed you, and with the activation key that is worth $400. Okay, we're going to give it to you. Then we'll also give you 10 pipeline tools for bioinformatics analysis worth $20. Okay, so this is just to give, it's just like a giveaway. All right, just like a giveaway, because this is the normal price we sell this course, but this is just at a very discount price. And after this workshop, is going to go. So in case you are interested, I believe you should be able to come, pay $6 to get access to the fundamentals of bioinformatics course, where you will learn phylogenetic analysis, multiple sequence alignment, molecular identification, right? Where you do molecular characterizations of bacteria, and then you learn how to do whole genome sequence. This is whole genome, not metagenomics now. Whole genome sequence phylogenetic analysis. You know how to do that, how to plot phylogenetic tree of any amount of whole genome sequences. Then the next thing we have is NGS data metagenomics workshop. Okay, so this is the workshop I'm talking about. We'll be working with WGS samples. All right, we'll be working with WGS samples. Okay, NGS data metagenomics workshop. So on the normal price is $200, but we are offering it right now at $25. So we are going to take you through the practical applications from data analysis down to taxonomic classification, down to AMR and analysis, down to visualizations of result for the research that you want to do. And if you go with metagenomics, we'll give you the fundamentals of bioinformatics for free. Okay, you will get this particular course for free. okay? And then we'll give you the paid software as well. We'll give you 50 pipeline tools for bioinformatics analysis. And then you have slots for mentorship. In case you have a research project, you want to discuss your research project with an expert at Genomark Hub, right? You want to discuss your project, you have challenges in your project, you have this access. So in 6 months time, during your project, you can discuss with us to tell you, to give you advice. Okay, why don't you do it this way? Why don't you do it that way? So it's like a free booking for you by the time you enroll in this metagenomic course. It is 100% practical. Because we have done most of the theory here, but everything you'll be doing in the workshop is 100% practical. Then we have research project to publish paper. So this research project to publish paper is on NGS metagenomic workshop. Is complete comparative and functional metagenomic study. All right, complete comparative and functional metagenomic study. I will show you one of the research papers that we did on metagenomic study. So this is like research project to publish paper. If you have a sample in metagenomics study, this is for you. Okay, and it's normally $800, right? But we're offering it right now at $200. Okay, it is $200 we are offering it, and then we are giving 85% waiver, okay, which is $120. On the website it was $200, but now we are taking $80 out. So here you have access to the fundamentals of mathematics, all courses on practical videos on metagenomics workshop, and then we'll go step-by-step practical approach, step-by-step practical approaches when it comes to the research paper. Okay, you are going to come up with a research proposal. We'll be giving you 100% practical guidelines. So let me even show you the research paper, like an example research paper that you make. I hope is this one. Hello, are you with me?
Yes, sir. Yes, sir.
Yes, yeah.
All right. So here's it. Exploring antibiotic resistant genes, mobile gene element, and virulence genes factor in urban freshwater sample using metagenomic analysis. Now, this is one of our students who was doing his PhD in China. All right. So what he did actually was he worked with public access data samples for this particular research. So he did resistant genes, taxonomy classifications, mobile gene element, virulence factors. Now, this is the published paper on metagenomics analysis. As you can see here, this is bioinformatics analysis process. We're going to take you through all these processes step by step. You will have a research paper like this at the research workshop. Okay, you will have a research paper like this at the research workshop. Okay, here as you can see, it has this is the methodology, metagenomic assembly and plasmid chromosomal sequence. Okay, so functional notation of MAGs. We have the data quality control and assembly assessment. Okay, we have the bacteria abundance and diversity assessment. We have the functional genome annotation. We have the ARG abundance and diversity assessment, as you can see here. Now, this is the metagenomics analysis that was done. There were more. All of those are still at the complementary, right? Then we have the abundance distribution of ARG types. So this is the kind of visualizations that will be done, okay, at the research project. This is for chromosomal plasmid unclassified.
We are not seeing what you are saying.
You're not seeing it? Are you not seeing my screen?
Yes, I can see
Yes, sir.
it. I
Yes,
don't
I can
know.
see.
Maybe that's network.
I can see.
Amazing. So we have the ARG abundance and diversity assessment. Okay. So these are some of the visualizations of results. This is what we will do in the research workshop, please. This is what we will do at the research workshop. Okay. This is the visualizations for antimicrobial resistant profiling. Okay. This is for the chromosomal plasmid unclassified sequence. This is another kind of analysis entirely, which is not found in the workshop. Okay, but every other one is found in the workshop, like the virulence factor, the resistant mechanisms of, and this is done comparatively. Can you see sample 1, 2, 3, 4, 5, 6, 7, 8? So no, like 7 samples here. Yeah, and 8 samples. So yeah, 8 samples. And it was done comparatively, right? It was done comparatively. This is the classifications. And this is the expression of the levels of MHs and MRGs. As you can see here, then we have the mobile gene element and meta-resistant genes. This is the network analysis, right? And this is discussion and conclusion. So if you want to have this kind of paper based on your study of interest, because we'll give you opportunity to work, okay? We'll give you opportunity to work with your data of interest, okay? With your sample of interest. You can even work with sample, be it clinical, whatever. And you have this research paper as your own. And another opportunity you have if you want to go with the research workshop is that there is mutual benefit. Other participants can also put your name in their papers if they are, because different participants should be able to work with different kind of metagenomic sample with different kind of focus, with different kind of research focus. Okay, so this is we'll provide you with the link for you to get this full detailed study material. But we have the outline for the research workshop, just in case you're interested. Okay, just in case you want to do that. This is the research, it's 2 months program. Research workshop is 2 months program. But the what's it called now? The metagenomics is 3 days, okay, for this, and then we give you 1 year access to the course so that you can go through it again. But here you have access to the videos for life at the research project, published, to publish paper. All right. So, but this one is 100% practical video. This is 100% as well, but it's just that it is research. This is research. This is just workshop. Okay, this is workshop, and this is research. All right. So, if you are interested, I'm going to drop at the chat room. Okay, if you are interested, so you just need to chat me up. Okay, I'm going to drop. Sorry, give me a minute. So, but before then, do you have any question on what we just showed you? Any question?
Hello.
Hello. Yes.
Hello.
Hello. Hello.
Yeah,
We are
can
with
you
you.
hear me?
I'm with you.
Okay. All right. Please, regarding the first package, this one, fundamental of bioinformatics, né? You said it's now $6.
Yes.
Then yes, you said here we can also learn whole genome sequencing, maybe annotation and whole genome sequencing in this fundamental of bioinformatics.
Yeah, whole genome sequence for like for phylogenetics, for evolutionary study. Evolutionary study.
Yeah, the whole genome sequence, not the ITS sequencing.
Yes, ITS and whole genome is there inclusive.
All right, fine. Then please, regarding my question, because I have seen you guys, you've been talking, I've been silent this morning, So I just have like 2 or 3 questions, please.
Okay.
Okay. Regarding metagenomics, please,
yes.
If you're talking about the sampling and the the DNA extraction, please, regarding metagenomics, are we just going to do the analysis from the sample, or we need to extract the DNA? Like, it's a bit confused, because I know for whole genome sequencing, we need to extract the DNA maybe of the microbe of interest, because that we need to culture them. We need to culture them on plate, then do the extraction, then we can now do the whole genome sequencing. But in case of metagenomics, do we do the analysis from the sample itself? Like, it's a bit confusing, eh?
The analysis from the sound is from this the genetic sequencing. It's after sequencing. Is that clear?
No, for metagenomics. Okay, let's assume I want to do the metagenomics for of fungi community in a feed sample. So am I going to culture the feed? Am I going to culture it on on a plate, or I will just do the analysis from the feed itself?
Okay. Culturing from the plate or the feed itself. Well,
yeah.
The thing is, you have to do, I think you have to do culturing. Hello, are you with me? Hello?
Okay, please, let me try to explain.
Yes,
yeah,
yes.
let me try to explain better. You see, for all genome sequencing, I know
I'm
very
trying
well
to
if I
get
want
your
to do all genome sequencing. Okay, let me try to put it in this way. Okay, let's assume I want to do the whole genome sequencing for a particular fungi species.
Okay.
With my experience, I believe I need to culture that particular, maybe after isolating that particular fungi from the feed sample. After the isolation of that particular fungi, then that's why I can now extract the DNA and send it for the whole genome sequencing. But in case of metagenomics, how do I go about that?
Okay, I get you now. Well, if you are doing with metagenomics, you need the genetic material. You need to extract the DNA, of course. So you have to do culturing so that you can be able to extract the DNA. You get it? You can make more research. Okay.
Then now you define metagenomics as what they call the community, the analysis of the what they call the community of all microorganisms present in a field in a sample. Then if I'm to extract a particular maybe fungi species, I will not be dealing with the whole what they call the whole.
Is it? Do we have anybody with this?
Yeah.
Wait, me, I'm a molecular guy. I'm not the lab guy. You get what I'm trying to say?
Hello.
So is there anybody who can help us with this?
Yes, I'm a microbiologist. Yeah.
Thank you so much.
Hello. Yes, I think if from what he's saying, if it's metagenomics and he needs to, look at the biodiversity in a feed sample, from what has been defined as metagenomics, all he needs to do is to now get the feed sample from where the representative DNA of organisms present will now be analyzed. And from there, now go on to do the further studies to be able to identify what organisms they belong to. You don't need to do culturing in metagenomics.
Amazing. Thank you so much. I'm a molecular guy. Okay.
Please, it means
I'm not a lab person.
Yeah.
So thank you for that.
So what is it? Okay, bro. Sorry, then. So what you say now is in metagenomics, I don't need to do culturing, then?
No, you don't need to do culturing. That's why it's metagenomics, community DNA. Hello, can you hear me? Hello, can you hear me?
I can hear you now.
Yeah, community DNA, you don't need to begin because if there are some organisms that are called viable but not culturable, right? So you cannot even culture them, but they could be present in that community. Now, with analyzing the general DNA of the whole community, you will be able to identify these organisms that you cannot possibly culture. And they could be relevant in the work that you're doing. So there's no need to culture when you're talking about metagenomics. You just use the sample, the environmental sample, in this case, animal feed or whatever the sample is, and then go ahead to extract the DNA, representative DNA of the different organisms, which you now take further to the database and so on to identify the organisms that have them. No need for culturing in metagenomics. You don't need to culture the sample.
All right. Okay, sir. Please, I have a question for you. Thanks for that clarification. Now, you said I don't need to call. No, yeah, it's clear, but I have another question for him, please. He said I don't need to culture, but I must get the representative DNA sequence. Then how do I do that without culturing, please?
That is the metagenomics now. There was a page from his.
I think the man is not understanding.
Exactly. Maybe hello. I think maybe the host should go back to that slide and explain him again. Yes, how you get the sample, you extract the DNA from the sample using a DNA isolation methods, and then now analyze to find out the different sequences, the organisms that contain them. From there, you'll be able to identify the biodiversity of the organisms that are in that sample. You don't need to culture. That's what metagenomics is. You don't culture and metagenomics. Don't start from culturing because you can't culture. There are some organisms you can't culture. So if you were to culture, you will be able to find out all the organisms there.
Hello, this is it.
Hello.
Yeah, please. Before I would like to say something, but before I proceed, I just dropped a link for in case you're interested in any of the courses, either fundamentals of bioinformatics or metagenomic workshop, or you are interested in the research program, okay? In the research program. I just dropped a link at the chat room. Just chat me up on WhatsApp. We'll reach out to you after then. So now let me just try to clarify something, because like I said, I'm not a lab person, like all this microbial biological culture and stuff. So but this is it, okay? Just like she said, if there is no culturing, that means you work directly by extracting the DNA from the sample itself. You understand, that is gotten from the feed. I hope that is clear. Huh? Hello? Please, am I audible?
Yes, sir. Yes, sir.
Yes,
Hello, am I audible?
Yes, yeah.
Thank you, thank you. So this is it. When you get, okay, for instance, you are working with water sample. Take the water sample that you have, right? You can take it to the lab. And what they will do is that they will extract the DNA. There's need for extraction of the DNA before PCR. All right? Because I have understanding when it comes to the molecular aspect. Okay? So first, get the sample raw. Now, if there's no culturing, it means they have to extract the DNA from the sample itself. So the extraction of the DNA is the metagenomic sequencing. But you have to know, is it shotgun metagenomic sequencing you want to do or 16S RNA? 16S RNA is just specific on 16S RNA genes. It's just specific on some genes. But you see, shotgun is based on wide numbers of genes, right? The whole genome sequence, genes that is presented in the entire metagenomic samples there, in the sample, okay, in the metagenomic sample. Is that clear? So get a sample. The first thing is, please try to make your research which molecular lab is in your country. For instance, if you're in Nigeria, you can call INCABA, okay? Tell them you want to do metagenomics and tell them how do you want to do that, or how will you do that? So they may tell you to bring sample to the lab. And when you take sample to the lab, bam, they, sorry, I'm speaking Nigerian now. So when you take sample to the lab, they will do the DNA extraction. Then from the DNA extraction, sequencing will begin, either from PCR, from primer design, stuff of 18 PCR sequencing. And then the metagenomics analysis now begins squarely after you get your sample. But first, for you to get your sample, get a molecular lab. Speak with them. This is what you want to do. They will take you through the process. And then you can speak to people like us who are into the in silico aspect of the molecular aspect of the work. All right, and then take it from there. So that is it. And if you have the skill, for instance, if you go through this workshop we're taking, you have the skill already. So you do the analysis yourself, you finish your project and you get it. For instance, there's a lecturer from University of Ilorin. She was doing a PhD in metagenomics. She do shotgun metagenomic sequencing from INCABA. We only got the sample, we did the rich processing, we do all the analysis that we're going to be teaching in the workshop like that, and then we do the report writing, we give it to her, she finish her work, and then that's it. So the same thing also apply to the papers that I just showed you. Is that clear, please? Hello?
Where we can join these websites, please kindly contact me from the email or in the Teams.
Please, I can hear you. Hello?
Yes,
Hello?
yes.
Yeah, what do you say, please?
I'm saying that from where we can attend that workshop, where you send the link, or on the email or on the Teams.
We can't hear you clearly. Can you type it, please? Can you type it?
Hello.
Can you type it? The next question, please. So, the next question.
Hello, sir. Good evening.
Yes.
I have a question on metagenomics. As you explained on the DNA analysis for metagenomics, I think that in my understanding, there is a lot of organisms from the environments that we take. So how can we differentiate the one which we are interested to work on?
Yeah, you narrow down your research, and that is why this workshop is all about. Okay,
yes,
that's why the workshop is all about.
Yeah, that is my the one question. The other question.
You can narrow down your research. You can narrow down your research at the taxonomy classification, at the community, at the taxonomy classification, the biodiversity aspect of the work. You can narrow down your research to a specific organism you are looking for at the species level. Okay, and then when you are doing AMR analysis like resistant genes, virulence factor, you can actually know which genes are associated to the specific organisms you are also interested in or you are looking for. Okay.
Okay. The other point I want to clarify, I need a clarification that is, for example, what is the direct implication of metagenomics for development of resistant genotypes?
Okay, no, for the research of resistant genotype or development, which one?
Yes, but the direct implication or application of metagenomics to develop resistant genotypes.
Okay, resistant genotypes?
In plant.
Okay. Now,
yes,
if I try to get you, because I'm trying to get your question. See, development, if you talk about development of resistant genotype, it could be probably you want to develop a database or you want to develop a pipeline tool, right? Online pipeline tools that are so many resistant genes that is specifically associated to the metagenomics analysis. Is that what you want to do? Or do you want to explore and discover resistant genes? Which one?
And I want to know that in the case of whole genome sequencing for specific crops, we can identify the gene that is responsible for the given trait, that is resistant for a given disease. We can explore in this way, but in the metagenomics, there is a analysis, the DNA analysis, and I mean that structural and functional analysis of the genome of the micro.
Okay, wait. I think I get what you're trying to say. Okay.
We are focusing on the microbes. It is different from the plant or the host we are going to work. So I want to know that.
Hello. Admin Q. Now, this is it. I understand you. I think I understand you now. See. Hello, can you hear?
The genomic analysis.
Hello? Please tell her I'm busy.
Yes, sir. Yes, we can hear.
Hello, can you hear me? Hello, can you hear me, please? Hello? I think I can't hear him. Maybe we lost him. I can't hear him again.
Yes, sir. Hear you. Sir, can hear.
Hello, can you hear me?
Yes, I can hear you now.
Okay, so I think I understand your question. So what I'm just trying to say in essence here is this, okay? Is that you can actually know specific genes, okay, that is associated to a particular bacteria of interest when you are exploring AMR or virulence genes in metagenomics. Okay, you can know because by the time you do the analysis, it's there in the workshop. Okay, by the time you do the analysis, you will know, oh, this particular gene is associated to this particular bacteria causing this infection. Did you understand?
Yes, I understand.
Yeah, you can do that in metagenomics. Not only in whole genome, you can do that because when you do metagenomics, it will show you that this gene is associated to this particular organism. Okay.
Okay.
Yes. So any more question? Now, like I said, if you are interested in this workshop, let me share my screen again. Hello, can you see my screen?
Not yet, please.
I believe you should be able to do that, to see my screen now. So please, if you are interested in any of this workshop, okay, Fundamentals of Bioinformatics, NGS Data Metagenomics Workshop, and then you have this for free, and then we take you to the step-by-step approach in analysis of the entire metagenomic sample, from the rich processing down to the last step in the workshop. It's 100% practical. It's $25, and these are the bonuses you have. So you have right now, I just dropped my link. Just chat me up right now in case you are interested in it. And if you're interested in the research workshop, okay, you can also signify. Okay, in the research workshop, you have all these too. You have more bonuses. You have more time to chat with us, to discuss with your project, and then you can work with your individual sample. Okay, then you can have a research paper of your own, because we are going to take you through step-by-step approach. This is 3 months. This is this is 2 months. This is 3 days analysis work, and then this is just a course that you can get, and you know, this is just self-learn. This one also is self-learn. We give you the courses, the videos you to learn, but we'll meet live session to answer your questions, like maybe in application to the research you're doing or whatever you're doing. And then this one is the research workshop. So just in case you are interested, so we just drop the link. Okay, I'm dropping the link again. Just chat me up on WhatsApp now. Chat me up on WhatsApp right away, and then we talk from there. Okay, just chat me up right away from there. We talk. I've showed you the research paper that was done by one of our students from China, and this is the project. This is it. My name is Oluwaseyi Olawale, and I'm also included. So okay, so this is the name of our student, and this is on metagenomics. This is the research on metagenomics. We do different kind of visualizations of result. We take you to step-by-step approach. You'll be able to do this on your own. Even after the research workshop, you can do for another sample with a different research focus as well. So if you are interested, please chat me up quickly right now before we go to the next question. I'm still here to take your questions. Okay, so go to the chat room and chat me up right away. Go on, please. Any more questions? I'm with you. Any more question?
Hello.
Hello. Yes.
Sorry, it seems you answered the question already, but like my network was really bad, so I couldn't hear you very well. So I want to get the clarification again, please. So you said with all genome sequencing, we can evaluate the antimicrobial resistance genes. We can also evaluate the virulence of the factors genes, the virulence factor or genes, with both whole genome sequencing and metagenomics.
Mm-hmm.
So yes. I'm just asking, yeah, we can do both. We can evaluate the antimicrobial resistance
and virulence factor at the same time. Yeah, at the same time. Yeah, definitely.
With both analysis, no?
Which analysis?
I mean, with both whole genome sequencing and what's it called, metagenomics.
Yeah, you can do from whole genome sequencing, you can do from metagenomics. So if you are here, you can also apply the same principle to whole genome sequence. The same principle, but not all. But you can apply the principle of resistant genes and virulence factor and mobile gene element to individual genome. Okay? Instead of metagenomic techniques. So, and then we can take you through that as well.
All right, thank you.
Yes. So once again, do not take this opportunity for granted, because actually this is really at a very discount price. Okay, and you know, for instance, the paper I just showed to you right now costs nothing less than $1,500, you know, at the end of the day. So just for the analysis and then setup. So if you have this, you see we're doing this for you at $120, then is a huge opportunity for you to take on. So and then you can also, if you don't have that capacity, go on with the metagenomics workshop. You know how to do the analysis. And some of those analysis, apart from taxonomic classification, you can apply to your individual genome as well, and then you get this, you know, similar results for this individual whole genome sequence as well. Okay. So once again, I'm going to drop, I believe you should be there. Yeah, so I just dropped my WhatsApp link. Just chat me up, and then would explore that. Now, meanwhile, before we go, we'll be having 4-month webinar series. Okay, we'll be having 4-month webinar series that we commence from next month. Okay, so we're having it once in a month for 4 months. We'll be bringing in experts from different from different locations around the globe to speak to us, to talk to us, show us practical applications and different aspects of biosciences and bioinformatics, genomics, machine learning, drug discovery. So stay put. We're going to send you email for you to register, and you see us around. So thank you, guys. Once more, if you still have a question, I'm here in the next 5 minutes. So thank you guys for attending this webinar. See you next time.
Can I ask you, are you still there? Can I ask you something?
Yes.
Yeah, please, you mentioned something about ITS sequences that will not cost more than 20,000 naira. That was what you mentioned the other time. So please, I'm currently in Ghana now. Please, is there any place you can recommend in Ghana where I can do the work, the ITS sequences?
Oh, well, I'm in Nigeria, but I believe you should be able to search and ask around where they do ITS sequence in Ghana. It's not something much. Some molecular labs should be able to do it. Okay, because it's very, very difficult to operate whole genome sequence in a developing country, but not ITS. Okay, not ITS. Not ITS.
Okay.
Yes.
Okay, thank you.
You're welcome.
Hello.
Yes, I'm here. I'm here.
Okay. Thank you for the webinar. Thank you for that interesting webinar.
Thank you so much.
Yeah, please. I have a question. Although I've connected, so I've just joined sent you a message on WhatsApp. I just clicked on the link.
Okay, no problem.
Yeah, please. I did metagenomics analysis some years ago. But I had issues with publication, so I had not published some of those work. So, but then over time, I noticed that there are some works on that, and some persons have done like work in that area. So I would want I need software. Like I want to repeat some of those analyses I did to enable me publish, not with the software I use them, but something newer that can make the same work up to date. Since I already have the data and I have the sequences, I know them, they're already in the chamber.
Okay, let me ask. Hello?
Yeah, I'm here.
So let me ask you, is it 16S or shotgun?
Yeah, 16S RNA.
16S RNA.
16S.
Okay, so what do you want to do in particular? I believe it should be biodiversity, right?
Yes, I'll do biodiversity, even the taxonomic. I want to redo the taxonomic. If there is a new software I can use to change some of the analysis I did to
okay, do you know the software? Do you know the software you used before?
I don't really know, but maybe I can still send you some of the work I did.
Okay, no problem. So let's talk personally on WhatsApp. Send it, let me see it, and then I will give you some insight on what to do.
Okay, so for the software you talked about, if somebody wants it, how will one get it?
Yeah, you just enroll in one of the courses. You can enroll with on. Okay, see, you can download the software, but you may not be able to use it because of activation key.
Yes.
Okay. Yeah. So to get the activation key, you have to enroll in one of the courses we are doing.
And when is the course?
Pardon? This is it.
When?
No, you can actually get it right away because we just give you coupon code to enroll. That's it. So you get it, and then the activation key will be there. Everything you need to know about the use of the software. I mean, the software for the analysis that is associated to the fundamentals of bioinformatics will be there. How you need to activate it to be there. How to install it will also be there.
Okay, but you talked about a course. When is the course coming up? The one you said one will have to enroll.
Yeah, which one do you want to enroll there? Is it the fundamental NGS or research project?
I think I might want to go in for all.
For all. Okay, so if you want to go for all, once you go with this, you get all this. You don't need to pay this again. Do you understand? You don't need to pay this amount. Both of this is free in this. So you just need to go with this, and then you get access to it. And also, since
Sorry,
you have
which one will I go with? Which one will I go with to get others?
now, based on what you said concerning the challenge you have, I would suggest you go with the research projects, because here you will be able to have a detailed insight on how you can actually improve your work beyond what you have already. Exactly. Yeah, beyond what you have already. So since you are using 16s, imagine maybe when the time comes you want to do shotgun, or you have a student, because looking at you, I think you're a professor.
I'm a lecturer then.
Exactly. So having a PhD student who wants to do metagenomics or shotgun metagenomics, you already have the experience, you already have the idea, you already know the tools to use. So definitely it will work out for you. So you can go with this, with the research project to publish paper. Because at the research project to publish paper, we will take you through the step by step, show you how you visualize your result, how you interpret your result, how you will compute your result for paper publication. I hope that is clear.
Okay.
Yes. So I would suggest you go with this.
Okay, so the one for 120 is for what?
This 120 is for research projects to publish paper. So it means that we'll be doing metagenomics. It's metagenomics. So metagenomics research workshop. Let me show you.
Hello?
I hope you get it. It's metagenomics research workshop. Can you see? Full-scale research on comparative analysis and functional metagenomics. Have you seen it? Can you see my screen?
Okay, so functional metagenomics.
Pardon?
You said that the 120 only is for what?
It's for full-scale research on comparative analysis and functional metagenomics.
Please, I have a short.
Please let me be through with her before you talk. Okay. Sorry. Go on, ma.
Sorry.
Hello, ma.
Sorry. Maybe please, I'll talk with you later. Hello?
Pardon? Hello, sorry, I can't hear you clearly. Hello, ma. Hello. Hello, Manchika.
Hello? Yeah, maybe your dad is coming, please. I mean, okay, you were saying something about the 120 on. Sorry, I had a call trying to cut off the call.
Oh, okay, all right. So go on. So I talked about $120 on. Here is it. It's a full-scale research on comparative analysis and functional metagenomics. Do you understand?
Okay. Work I did, I also did some things on functional genes. I did some functional specific genes too.
Exactly. So it means that you can use your sample. Okay. It's possible to use your sample to walk through this process. Okay, you can use a sample to do the same thing. So it means in the research workshop, you'll be working with your sample to do your paper, because the essence of the research workshop is to have a paper at the end. You have a research paper at the end.
Okay, I think I'll communicate with you more online, since I've just sent you a text on your WhatsApp.
Okay.
So I think you have to do a follow-up there.
What's your number again? Your number?
It ends at 82, 882.
882. I can see that. 4882.
Yes.
Okay. Okay, good,
Right.
great. So I would suggest you go with the research workshop, because it will help you. You already have a sample. Okay, so the improvement of the paper, working on the paper, doing all of these things, because all the analysis you need to know to do is there. Okay, the step-by-step is there. The how to compute your result will be there. How to interpret your result for paperwork.
What of the research and the other projects you talked about?
Which other project, ma?
No, I think there are about 3 types. The other one you said
this is, yeah, when you go with this, okay, when you go with this, you will have this course, and then you have this course. But we'll be using some of the things in this course, or all of the things in this course, and more that is not there, to do the research project published paper for metagenomics. That's what I'm saying. So this one, this.
Okay, don't worry. Yeah, okay. Let me continue the start online. Maybe I'll give you time. Later we'll talk.
Great, great. No problem. No problem. No problem. So I think someone else has a question. Pardon?
No, not you. Thank you.
Okay, please, can you mute then? Thank you. So somebody else want to say something? Who is that?
Yeah, please. Thank you so much for the informative webinar. I have 2 questions. The first one, currently I'm not working in metagenomics, but I'm interested more in plant genomics and data analysis. So my question, which one of these courses is most fit to me? The first one or the second one?
So what are you doing?
I'm interested more in plant genomics and data analysis. So I'm working in biotic stress, and also I do some plant transformation. So I'm interested more in lab work and also plant genomics. So which one of these is the most fit to me, my interest?
Okay. I wouldn't suggest you to do this metagenomics since you are working on plant genomics. So I want you to work. I want to suggest you a different package. We have a different package. Where are you joining from? Sorry.
Originally I'm from Egypt, but now I'm currently working in Canada.
Okay, great, great. So can you chat me up? I will give you a different package for plant genomics.
Okay, sounds good. Okay, the second question is about the class itself. Should I attend this at specific times, or you send a recorded webinar, or because the time difference between Canada and Africa?
Sorry, can you say that again?
Are you sending, regarding the classes, are you sending like a recorded classes, or I have to attend at specific times, because there is a big time difference between Africa and Canada?
Okay, is it for the package I'm going to show you, or for this?
Is it again?
Is it for the package I want to show you, or you want to, or for the metagenomics? Do you also want to study metagenomics? Do you want to learn metagenomics analysis as well?
Yeah.
Okay, because you need it. We have a package for plant genomics. Okay, we have a package for plant genomics, and we also have a package, bro. On the gen. We also have a
Let's know about the plant genomics, please. We are also interested.
So you have the same discount for the plant genomics?
Yeah, listen. I'm trying to sort, to answer your question. You have interest, right, in metagenomics, isn't it?
Mainly in plant genomics and data analysis.
Okay, so can you just chat me up? Okay. Just chat me up.
Sure, I will.
Because we have video courses, so we can give you the practical videos courses, and then you just join for to ask questions and answer to clear doubts, to clear your doubts and things like that during the workshop. Okay. So, and if it is research workshop also, we have practical videos. Okay. We'll have, after the session, we'll send you the videos. You do your part, you send your work for review and things like that. So it's not necessarily until you are available. We can actually send you video courses that you can have. Is that clear?
Yeah, nice, good.
Yeah. So just chat me up. Let me drop my. I think just check the chat.
I already sent a text. Yeah, I already messaged you.
So what's your number again? What does it end with? 4501, right?
Right. Yes.
4501 from Ghana.
Yeah. Right. Yeah. Yeah. From Canada.
From Canada. Sorry. Canada. Yeah. Okay. So we'll talk. Okay. Don't worry. We'll talk in details. So who else? Who else, please?
They are asking questions now.
Hello, go on.
Hello. Please, which genotyping company is cost effective and repeatable?
In what?
Which genotyping company or sequencing company is cost effective and repeatable as far as you know?
Sorry, I can't get your question. Is it possible you type it?
I mean that which genotyping company or sequencing company is cost effective and repeatable, especially in Africa.
So can you type it? You are breaking. Okay. I can't hear you clearly. The network is breaking from your end.
Okay.
Hello, you get me?
Hello, can you hear me?
Yeah, can you just type it and send it to the chat room? I would appreciate that.
Okay.
Who else has a question? Hello, who else has a question before the typing comes in? Our course is recorded, okay? Our course is recorded. You will definitely get the recordings, okay? Our course is recorded. So just chat me up. Yeah, we're going to provide a certificate for the webinar. Okay, no certificate for the webinar, please. We are going to provide certificate for the webinar series that is coming up next month for 4 months, okay? We're having a webinar series, experts from different places around the globe that will take you on, okay? Please. Hello, I'm still waiting for your question.
Daddy, your question. No.
Hello. I'm waiting for your question. And if that's all, so thank you guys. Hello, is there more question? Or that's all? So let me introduce the course again. So I said we're having a course. We're having courses, and then we're having workshop, and then we have research workshop. Okay. So the first course is $6 at 88% waiver. So if you're interested, just check the chat box. You see my WhatsApp number. Chat me up, okay, to get access to it. And then for those that want NGS data metagenomic workshop, all right, we'll give you this for free, and then you have 87.5% discount. So you'll be, instead of $200, you're paying $25. And these are some of the bonuses that's available, like 50 pipeline tools for bioinformatics analysis will be provided. And then we have research workshop on metagenomics, okay? Research workshop, research related to published paper. You have a paper at the end of this program, okay, on metagenomics. And then you have this for free, you have this course for free, okay? It's $800, but we are giving 85% waiver. So you just pay $120. In case you're interested, we'll give you 100 pipeline tools, not 50, 100. And then you have paid bio software tools, 2 bio software tools. So if you're interested, check the chat room, chat me up. Okay? Send the chat and then we'll reach out to you.
Hello, mommy. Ṣé ní malaria ni?
Àwọn ò ní ní malaria o, ẹ má fún wọn malaria o. Ibo lẹ ti ń sọ̀rọ̀? Àti bo Ìbàdàn, Ọ̀yọ́, Ìlọrin. Hello?
Hello.
Hello.
Can you hear me?
Yes, yes, yes.
If somebody want to pay, is he paying, for instance, somebody in Nigeria, will he pay equivalent of dollars or it must be dollars?
No. It's equivalent in naira actually, in naira. So you just calculate, okay?
So what? Anyway, we'll talk on WhatsApp anyway.
Okay, you already chatted me up, right?
Yes.
What's your number
yes.
again? What's your number? What's it ending with?
88.
88. Can you start? Okay, I've seen it. I've seen it. All right, no problem. No problem.
Have you seen
we
it?
talk. Yeah, I've seen it. No problem. We talk. So thank you guys. This is the end. Don't forget, we'll be having webinar series, 4-month webinar series where we'll be having expert from next month. So we'll be doing quite a number of different things. It's going to be transitional, actually. So thank you. See you guys next time. Bro.